Ferns, belonging to the group Polypodiopsida or Polypodiophyta, are vascular plants lacking seeds and flowers, reproducing through spores. Distinguished from non-vascular plants by their specialized transport bundles for water and nutrient conduction, ferns exhibit life cycles where the branched sporophyte phase is dominant. With intricately designed leaves known as megaphylls, ferns exhibit a higher complexity compared to club moss's microphylls. The majority of ferns fall under the category of leptosporangiate ferns, unfolding from coiled fiddleheads into fronds. This diverse group encompasses approximately 10,560 known extant species, comprising both eusporangiate and leptosporangiate ferns, with the former including horsetails, whisk ferns, meradioid ferns, and ophioglossoid ferns. The fern crown group, tracing back to around 423.2 million years ago during the late Silurian period, marked a crucial moment in the evolution of land plants. Some fern species like bracken and water fern are problematic weeds globally, while others like azolla contribute significantly to nitrogen levels in rice paddies. Extant ferns, in their sporophyte form, typically do not exhibit woody growth, with any woody elements mainly found in the stem. Their foliage varies between deciduous, evergreen, or semi-evergreen, depending on environmental conditions. Similar to seed plants, fern sporophytes are comprised of stems, leaves, and roots, and unlike spermatophytes, they reproduce through spores rather than flowers and seeds. Ferns are distinguished from spore-producing bryophytes by being polysporangiophytes, with sporophytes that branch out and develop multiple sporangia independently from the maternal gametophyte. The green photosynthetic section of a fern is known as a frond, technically classified as a megaphyll. As new leaves grow, they unfurl from a tight spiral called a crozier, or fiddlehead, into fronds, a process known as circinate vernation. Fern leaves are categorized into sporophylls, responsible for spore production, and tropophylls, which do not. Spores in ferns are contained within sporangia, often grouped together to create sori, possibly shielded by a protective layer known as an indusium. The pattern of sporangia arrangement plays a crucial role in the classification of ferns. Fern species can be monomorphic, where fertile and sterile leaves appear identical and can photosynthesize equally, hemidimorphic, with only a section of the fertile leaf distinct from the rest, or dimorphic, holomorphic, featuring visibly different fertile and sterile leaves. Fertile leaves in dimorphic ferns are usually narrower than sterile ones and may lack green tissue entirely, as observed in certain fern families like Blechnaceae and Lomariopsidaceae. The anatomy of fern leaves displays a range from simple to highly divided structures, with examples like Gleichiniaceae and Lygodiaceae exhibiting indeterminate forms. Divided forms can be pinnate, where leaf segments are entirely separate, or pinnatophid, where segments remain partially connected. Furthermore, branching of fronds can result in a blend of pinnatophid and pinnate shapes. Fronds are classified as bipinnate when leaf blades divide twice, tripinnate when they branch thrice, and extend to more complex structures like tetra and pentapinnate fronds. In tree ferns, the primary stalk, stipe, connecting the leaf to the stem, often features multiple leaflets. These leafy structures, called pinnae, may further divide into smaller pinnules. Although fern stems are commonly referred to as rhizomes, they grow underground in only select species. Some ferns, like epiphytic varieties and certain terrestrial ones, produce above-ground creeping stolons, such as the Polypodiaceae. Additionally, many groups develop above-ground semi-woody trunks like the Kythiaceae, known as the scaly tree ferns. In some instances, these trunks can reach towering heights, with certain species like Kythia brownie on Norfolk Island and Cyathea medullaris in New Zealand growing up to 20 meters, 66 feet tall. Fern roots, essential for absorbing water and nutrients from the soil, are subterranean non-photosynthetic structures resembling the fibrous roots of seed plants. In vascular plants, the dominant phase in the life cycle is the sporophyte. The gametophytes in ferns differ significantly from those in seed plants, existing as independent entities resembling liverworts. Unlike seed plant gametophytes, which rely on the parent sporophyte for nutrition and develop within the spore wall, fern gametophytes are free-living. A typical fern gametophyte comprises a photosynthetic, one-cell-thick structure called a prothallus, usually heart or kidney-shaped, ranging from 3 to 10 mm long and 2 to 8 mm broad. This prothallus generates gametes through antheridia, small spherical structures producing flagellate antherozoids, and archegonia, flask-shaped entities yielding a single egg at the base. To anchor the prothallus to the soil and absorb water and minerals, root-like rhizoids, which are elongated single cells, play a crucial role in the fern gametophyte structure. The life cycle of a fern is characterized by two distinct stages, akin to club mosses and horsetails. Initially, spores are generated by sporophytes within sporangia, grouped in sori, singular sorus, located beneath fertile fronds. Subsequently, these spores germinate into a brief structure known as a gametophyte, supported by rhizoids and firmly rooted in the soil, responsible for producing gametes. As a mature fertile frond hosts sori and releases spores, these spores land on the ground, take root through rhizoids, and mature into a prothallus. The prothallus features rounded antheridia, which yield antherazoids, male gametophytes, and archegonia, producing a single oosphere. The antherozoid navigates the archegonium to fertilize the oosphere, leading to the formation of a zygote. This zygote transforms into a distinct sporophyte, while the gametophyte continues to exist as an independent plant for a short period. Carl Linnaeus in 1753 established the classification of ferns and fern allies, recognizing 15 genera under the class Cryptogamia, dividing them into felices, such as Polypodium, and musci, mosses. By 1806, the number of genera had expanded to 38 and continued to grow over time. Initially belonging to the class Felices, ferns were later placed in the Pteridophyta or Felicophyta division of the plant kingdom, which is no longer valid due to being paraphyletic. The term Pteridophyta became synonymous with ferns and their allies, but led to confusion as it was also used for a specific group within the division. Traditionally, ferns were categorized into three main groups: eusporangiate ferns like Ophioglossake and Maradiaceae, and leptosporangiate ferns. Other plant species like club mosses, spike mosses, quillworts, whisk ferns, and horsetails were considered fern allies. Genetic studies revealed relationships among these groups, with Lycopodiophyta being distantly related, while whisk ferns and horsetails are closely linked to leptosporangiate ferns. Molecular phylogenetics revised the classification, placing ferns under manilophytes within the Tracheophyta division, alongside seed plants and lycophytes. The inclusion of Equisetaceae in ferns was supported by molecular and morphological evidence. Leptosporangiate ferns, often termed as true ferns, encompass most familiar fern species, with the Osmundaceae family considered an intermediate between eusporangiate and leptosporangiate ferns. Despite these classifications, uncertainties persist in understanding the relationships within major fern lineages. Equisetaceae's placement within ferns remains a topic of debate, with suggestions to elevate Ophioglossales to a separate class, distinct from other fern groups. Lycopods have previously been placed in subclass Lycopodidae, while ferns were divided into 5 subclasses: Equisetidae, Ophioglossidae, Celotidae, Maratiidae, and Polypodidae. Christenhuss et al. In 2011 followed this subclass division and recombined Smith's Psilotopsida as Ophioglossidae, maintaining 4 subclasses of ferns. Ferns are widely distributed, with the highest concentration found in tropical regions and the lowest in Arctic areas, with the most diverse array present in tropical rainforests. New Zealand, recognized for its association with the fern, boasts around 230 species spread throughout the country. Ferns often excel in challenging habitats where conditions limit the success of flowering plants. Some fern species, such as bracken fern in Scottish Highlands and mosquito fern in tropical lakes, are considered invasive weeds, forming large and rapidly spreading colonies. Ferns can be found in 4 main habitat types: moist, shady forests, rocky crevices shielded from intense sunlight, acidic wetlands like bogs and swamps, and tropical trees where many species grow as epiphytes. Epiphytic ferns, in particular, harbor a diverse range of invertebrates, with some estimates suggesting that bird's nest ferns alone support up to half of the invertebrate biomass in a hectare of rainforest canopy. Many fern species rely on symbiotic relationships with mycorrhizal fungi for their growth. These plants display specific pH requirements, like the climbing fern in eastern North America thriving only in moist, highly acidic soils, while the bulblet bladder fern prefers limestone habitats. Fern spores are a rich food source containing lipids, protein, and calories, leading some vertebrates to consume them. For example, the European wood mouse, bullfinch, and the New Zealand lesser short-tailed bat have been observed feeding on fern spores. Ferns reproduce through spores and have a unique life cycle known as alternation of generations, including diploid sporophytic and haploid gametophytic phases. The fern life cycle involves a diploid sporophyte producing haploid spores through meiosis, which develop into a free-living haploid gametophyte by mitosis. This gametophyte generates gametes and fertilization results in the growth of a diploid sporophyte, representing the typical fern plant structure. Sometimes a gametophyte can exhibit sporophyte characteristics like roots or sporangia without developing into a full sporophyte plant. Various fern species like Pteridium equilinum, bracken, Mateuccia struthiopteris, ostrich fern, and Osmundastrum cinnamomeum, cinnamon fern, are valued as food sources, with fiddleheads being utilized in dishes such as budu pakas in Brunei. In New Zealand and the South Pacific, the traditional consumption of tubers from the para fern, Tisana salicina, king fern, prevails. Historical records indicate that fern tubers were a dietary staple in Europe 30,000 years ago and were used by the Guanches in the Canary Islands to produce gofio. While some ferns are carcinogenic, most ferns are generally not toxic to humans. Licorice fern rhizomes were historically chewed by Pacific Northwest natives for their taste. Notably, certain fern species like Azala, also known as water fern or mosquito ferns, serve as biological fertilizers in Southeast Asia's rice paddies due to their nitrogen fixation capabilities. The resilience of ferns against plant-eating insects is demonstrated through genetic engineering efforts, such as transferring the TMA12 protein-expressing gene from the edible fern Tecteria macrodonta to cotton plants, making them resistant to whitefly infestations. In horticulture, ferns are cultivated as ornamental and landscape plants, with popular choices including the Boston fern, Nephrolepis exaltata, the bird's nest fern, Asplenium nidus, and various Platycerium fern species. Perennial fern varieties find favor among gardeners in the Northern Hemisphere. The discipline focused on the study of ferns and other tridophytes is known as pteridology. A pteridologist therefore specializes in the examination of pteridophytes, which also encompasses more distantly related lycophytes. During the Victorian era, there was a phenomenon known as pteridomania, characterized by a craze for collecting ferns and incorporating fern motifs into various forms of decorative art. This trend saw fern designs adorning a wide array of items, ranging from pottery, glass, metals, textiles, wood, printed paper, to sculpture, appearing on everyday objects such as christening presents and even gravestones and memorials. The popularity of cultivating ferns indoors gave rise to the invention of the Wardian case, a glazed cabinet designed to safeguard plants from air pollutants and maintain the required level of humidity. Beyond decorative purposes, ferns found their way into other realms of application. The Barnsley fern, a fractal pattern created using a chaos game through an iterated function system, is named after the mathematician Michael Barnsley. This self-replicating structure is generated by a mathematical function iterated at various scales to produce a fern-like pattern. Additionally, dried ferns were utilized in artistic endeavors, either as stencils or directly inked to create designs. Noteworthy in this regard is the Ferns of Great Britain and Ireland, an example of nature printing wherein artist and publisher Henry Bradbury patented a method that transferred specimens onto soft lead plates. The ferns of Great Britain and Ireland served as an exemplar of this nature printing technique. In the realm of folklore, ferns hold significant roles, featuring in myths and legends surrounding mythical flora and seeds. For instance, in Slavic folklore, there exists a belief that ferns flower once a year during Ivan Kupala night. Discovering a fern flower is said to bring happiness and wealth for life, despite the flower being notoriously elusive. Similarly, Finnish tradition implies that locating a blooming fern seed on midsummer night grants the finder the ability to be guided invisibly to the sites where eternal will of the wisps, known as Arnevalkia, mark hidden treasures. These locations are enchanted by a spell that only the possessor of the fern seed can decipher.